In this part of the lecture, we will be discussing about the glomerular size and the formation of the loop of Henle. So this is a very important part of the topic as we are discussing the evolution of the kidney in the vertebrates. Now, here, first of all, I would like to give you some concepts that why there is a change in the glomerular size of the animals. So previously we have discussed that the glomerulus has evolved during different stages. So first it was the aglomerular stage, then we had the internal glomerular stage which was of primitive type. Then we had the modified type of the internal glomerulus. So after the formation of the glomerulus, now we have the change in the size of this particular structure. Now, why there is a change? Why the sizes of the glomerulus they change with the, you can say, with time? So, students, the adjustments to the existence in different environments, they have changed the size of the glomerulus. So, this is a very, very important thing to note, I think. So, if a particular animal is living in hypotonic conditions, or you can say if an animal is living in fresh waters, so what will happen that more of the water is coming inside that particular animal. So if I take a particular animal, suppose this is the animal, and what will happen if it is living in freshwater environments, so what will be happening that the water will be coming towards the inside of this animal. And this is a simple process of endoosmosis. So if you have low of the salt concentration outside the body, so the water will be coming towards the inside of the body. So this is endosmosis. So if more of the water is coming inside the body, and in these cases, what will happen? What is the need of the situation? So the need is to give out this water which is coming extra inside the body. So this particular animal which is living in fresh water will give out the extra water. And the nephrons or the kidneys of this animal will be giving out a dilute kind of urine. So for making more of the urine, more of the filtrate is required by the kidney. So that simply means that if you have a kidney, suppose you have a nephron in that particular kidney. So I'm just making a nephron. This is the loop of Henle, and this will be the DCT and PCT. This is the collecting tubule. And here I am just drawing one glomerulus, which is present over here. So if this particular nephron has to give out extra water, what is the need? So we need the formation of more filtrate in this particular section. So we need more of filtration, we need more of ultrafiltration, we need more of primary urine. So this is the mode of excretion in this particular animal. So in these cases, what has happened that the size of the glomerulus, it has increased. Now why it has increased? To make more of the filtrate. So if you have more of the filtrate made, there will be more excretion of the water which will be going out of the body. And that's what we needed. And if we have a situation which is opposite to this particular condition, and if a particular animal is living in a hypertonic kind of environment, what will happen? The water from the body, it will go out. So what the animal will need? It will need to conserve the water. So if we have to conserve the water, what will happen? The glomerulus size will decrease. So if you have, you can say, a narrow kind of glomerulus, small glomerulus, a small filtrate will be formed and less of the urine will be given out. So that means the size of the glomerulus, it can increase or decrease the filtrate which is produced in the nephron. So that is a general kind of, you can say, sentence that if you are living in hypotonic or freshwater conditions, you will be having bigger glomerular size. And if you are hypertonic, you will be having smaller glomerular sizes. Now students, apart from this particular thing, there are some other mechanisms by which an animal can develop resistivity to the incoming and the outgoing of the water in these particular environments. Now, if I take one particular situation, let's suppose an animal, it is living in hypotonic solutions. So that means more of the water is coming towards the inside of the body. Now, if the water is coming towards the inside of the body, what will be the glomerular size? It will be bigger in size. Other to the glomerulus, what the animal can do? Number one, it can make a copious or a mucus secretion over the skin. So what will happen if it will secrete a mucus layer over the body? What can happen that less of the water will be coming towards the inside of the animal. So that can be done by the animal. So that was the first thing that animal can, by which the animal can resist the entry of the water. Number two, it can be the reduction of the integumentary blood supply. So this is again a very, very important thing. If you can reduce the blood supply in the skin, the intake of the water or into the blood, it can be reduced. So that is seen in all the fishes which are living in hypotonic kind of solutions. Now students, here one thing is to be mentioned, that suppose if an animal is secreting this mucus sheet, and this is so, you can say, efficient that it can stop the entry of the water into the body. Now, does this animal require a large or a bigger glomerulus now? So my answer will be no. And that is what has happened in the mammals. I'm not talking about all the amniotes, or you can say the birds or the reptilians, but I am talking about mammals. Now in mammals, what has happened that they need to conserve the water. As they are terrestrial, they need to conserve water, but they have no signs of the glomerular change in them. So I'll be taking about this particular topic later on. So the second thing what the animal can do is the reduction of the integumentary blood supply. Now the third thing what this animal can do is that this animal can make some kind of thick body armor over this whole body, just like this. It can make a thick body armor which can decrease the intake or the incoming of the water. So this kind of body armors, they have been seen in placodermi kind of fishes. So there is a group of fishes which is called as placoderms. And we have another group which is called as ostracoderms. So the placoderms, they had plate-like bony armors over their chest or over their whole body. So this bony armor was very, very important to stop the incoming waters. So that was the third thing that these animals can do. Now the fourth thing is the increase in the size of the glomerulus, that what we have discussed over here. So this is the fourth kind of a thing which this animal can do. So students, these are the, you can say, the adaptive measures that a particular animal can go for the, you can say, removal of the extra water. So that was some concept of the glomerular size increase in the animals which were living in the hypotonic medium. Now, if we go for the animals which are living in hypertonic solutions or the hypertonic media, what will they do if they are living over there? Suppose this is the animal which is living in the hypertonic medium. So what will be happening? That more of the water will be going out of the body. Now they have to conserve this water. They have to take this water in. They have to take more of the water in because their body water is going out. So they need to conserve this water. And what will happen to the glomerulus size? That the size of the glomerulus will decrease. So there will be a smaller glomerulus in this particular case. Now, apart from this glomerulus size, what this animal can do? Number one, this is the very same thing which was happening in the previous case, that there will be a formation of, or you can say a secretion of the mucus over the body. So that can reduce the outgoing water. So it can help in conserving the water by this particular animal. Now the second thing which is very, very important, it is again the reduction in the integumentary blood supply. So if you can reduce the blood supply in the integument, you can again reduce this outgoing water quantity. So I would like to say that the first two remedies, they are very similar in the hypotonic and the hypertonic conditions, both. Now, if I go for the third strategy that can be taken up by these animals, it is the decrease of the glomerulus size. So this will be third strategy, to decrease the size of the glomerulus. And you can see this kind of situation in the, you can say, the bony fishes or the teleost fishes. So they have reduced glomerular sizes. If I go further, you can say they may increase the bodily osmotic pressure. Now this is again a strategy to cope up with these kind of situations. So this is the fourth strategy which they can increase inside their bodies. So if they increase the bodily osmotic pressure, they can reduce the outgoing water. Number fifth, what they can do is the excretion should be by the gills, and that should be of the salts. So the excretion of the salts, they should be by the gills, and they should be extracted from the ingested seawater. So what will happen? That if you are retaining more of the salts inside your body, that will give you more of the water. So extra salts can be removed by the gills. So that will be a good important thing. So that these animals can do. Number 6 can be the excretion of the salt by the glandular tissue around the external nares. So you can give out the salts from the salt glands, as we have seen in the cases of the birds or the marine birds. So they are giving extra salts through the salt glands. So they are conserving water in this particular case. Now, if they have to remove that salt from the kidney, that required some water. But if these salts are removed by the salt gland, they can remove it in a very concentrated manner. So that can conserve some of the water inside the body. So that is again a strategy. The next strategy can be the excretion of the salts in a hypertonic urine. So this is again a strategy of this particular animal, that if it is living in hypertonic conditions, it will give off a hypertonic kind of urine. So that is again conserving the water of the body. Now if we go further, the excretion of the salts is taken up by the lacrimal glands in the marine turtles. So this is a kind of a special kind of excretion in the marine turtles, that they are giving out the salts by their lacrimal glands of the eyes. So by these kinds of strategies, they are coping up with the hypertonic kind of environments. So students, here what we are discussing is the glomerular size. So it can increase or decrease by the environmental conditions. Now students, we are to the next part, which is the loop of Henle. So here I would like to give you first of all some concept that why this loop of Henle has developed in the animals. In a particular nephron, what we had is a Bowman's capsule, and we have a PCT, we have a loop of Henle, and we have a distal convoluted tubule, then we have a collecting tubule. So that is the structure of a typical nephron of a mammal. If we go in the formation, or you can say the evolution of these renal tubules, what we discussed in the very first lecture, we had a peritoneal funnel, and it had a connection with the Bowman's capsule, and then it was having a outpour just like this. So that was the case when this particular nephric tubule was formed in the evolution. So here we had the glomerulus over here, but it had a connection with the splanchnocoel of the animal. So what we have discussed in the evolution of the renal tubules. Now after that, what has happened, we had a tubule just like this. We had a Bowman's capsule, and we had a tubule just like this that was opening into a particular duct, and that was the archinephric duct or the holonephric duct, which we have discussed earlier. Now what is the point? That this particular structure in the center, which is said to be as the uriniferous tubule, or this part of the tubule, it is the uriniferous tubule. It has developed so much that it has been extended to, I should say, this part. So it has been attributed to the formation of the proximal convoluted tubule, then we have the loop of Henle, then we have the distal convoluted tubule, and we have the collecting tubules at the very end. Now this is a kind of, you can say, the evolution in the capillaries of the nephron. What has not changed is this particular part. It is the Malpighian corpuscle, and this is present in all the three sections. Now what is the difference? There can be a size difference, there can be some difference in the epithelial membranes, but they are still okay, they are still fine, and they are not changing as per the histology is concerned. But sizes can vary, as we have discussed earlier, of the glomerulus, and the sizes of the tubule, or the, you can say, the capillary or the capillary network, it has also changed. Now why there is a need to make a loop of Henle? Now this is the most important thing, and how they have developed. If we go for the conditions of the animals, as we have discussed previously in the glomerular size topic, if you have a hypotonic kind of environment, you will be having a big glomerulus. If you have a hypertonic kind of environment, you have a small glomerulus and a small Bowman's capsule. In mammals, in this particular case of mammals, what has happened that these animals, they need to conserve the water really. So that is, they are living in a dehydrating kind of environment. So they are actually conserving the water. But what has happened that their glomerular size, if we go for the size of the glomerulus, and if they are in the dehydrating conditions, it should have been reduced to this much of size. A very, very small glomerulus they should have. But they have retained a proper size of the glomerulus. They have not decreased the size of the glomerulus. But what they have made a loop of Henle instead. So what this loop of Henle will be doing, I should go with the mechanism first of all. If I go back, first of all, I should go back. Suppose this is a particular animal in which we are talking about the hypertonic conditions or the dehydrating conditions. So what this animal will do for coping up with the dehydration conditions? So it should give out a concentrated urine first of all. Now this urine should be concentrated. For making a concentrated urine, what should be small? The glomerular size should be small. But here the glomerular size is not small and it is quite big in size. But here the concentration of the urine is taken out, or it is taken by this loop of Henle. So it is concentrating the urine to such an extent that not even an ml of extra filtrate is given out in the urine. So students, this is the main, you can say, the basic concept of why the loop of Henle has been developed in mammals and in amniotes. So this is to concentrate the urine, and this is the main mechanism of the loop of Henle. So it is also called as the hairpin loop because of its structure. So it is just like a hairpin. That's why it is called just like this. So in mammals, the glomeruli, they have remained in the larger sizes. So because of this particular technique of water conservation that is taken up by this hairpin loop or the loop of Henle. So in general, if I speak about the hairpin loop sizes, if you have, you can say, a concentrated kind of environment or a hypertonic kind of or a dehydrated kind of environment, this loop of Henle will be bigger in size. And if you have a hypotonic kind of solution, so there is no need to concentrate the urine, so this loop of Henle will be decreasing in size. So the length of the tubule will decrease in size. So conclusively, we can say that the sizes of the glomerulus and the size of the loop of Henle, they can increase or decrease according to the environmental conditions, and thus we have different types of nephrons in all the animals. So students, I think that was all about the particular topic of the glomerular size and the loop of Henle's. Thank you.